This video will be talking about the Parkinson disease and how the recent discovery of master regulator gene being the cause of such disease. Parkinson disease is a progressive nervous system disorder that affects movement. It affects people's walking, talking, mental and behavioral state, sleeping behavior, and memory. Well-known example of Parkinson disease patient would be Muhammad Ali, the greatest boxer of all time. Parkinson disease occurs because neurons that control movement become impaired or dies. Reduced number of neurons decreases the production of neurotransmitter called dopamine, because neurons are the source of dopamine. Scientists knew that neurons' death was causing Parkinson's disease, but did not know what was causing the death. Until recently, a group of researchers found that master regulator genes, specifically TT2, might be the cause. TT2 is a master regulator gene that is responsible for managing the types of chemical marks that annotate DNA and affect gene activity. It is heavily involved in important gene activities like cytosine modification and DNA methylation. Researchers believe that overactive altered TET2 were linked to epigenetic dysfunction, which heightened the immune response, which caused inflammation, and reactivated the cell cycle, which caused neurodegeneration to occur. Researchers found out that TT2 was the cause of the disease by looking at the bisulfite sequencing of each Parkinson's disease patient and control group. Bisulfite sequencing is the use of bisulfite treatment of DNA before routine sequencing to determine the pattern of methylation. The bisulfite sequencing enabled researchers to recognize that Parkinson's disease patients had a normal increase in cytosine modification enhancer as well as increase in hydroxymethylation. They also used RNA sequencing data to investigate transcriptomic alterations in the Parkinson's disease patient's prefrontal cortex. To confirm and test their hypothesis that TET2 loss will protect the neurons from inflammation and degeneration, researchers injected lipopolysaccharide, also known as LPS, that causes neurodegeneration to mice with active TET2 and mice with inactive TET2. A year after injection, the researchers found that mice with inactive TET2 did not experience neurodegeneration, whereas the mice with inactive TET2 experienced neurodegeneration. The discovery of TET2 as possible prevention of Parkinson's disease is a world-changing finding. This biotechnological method has not been fully proven nor tested on human genome yet. But if the use of TT2 to prevent Parkinson's disease was introduced to the public, it can prevent many sufferings. Every year, 60,000 new Americans are diagnosed with Parkinson's disease. The methods to slow down the disease exist, but no one knew about how to prevent such life-changing disease until now. The prevention of the Parkinson's disease by altering the TT2 will not only prevent many Americans from long-term suffering, but it will also contribute to the future research of other diseases, like Alzheimer's, by offering researchers a clue to what is causing inflammation and degeneration of neurons.